Hello, I'm Nate Beam, and I'll be presenting the extremophile Thermus aquaticus. I'll mainly be focusing on the Y51 MC23 and YT1 strains, which I will call Y51 and YT1 for short, but also add supplemental information about other closely related thermophiles, such as T. scadoctus, which I will call TS for short. Thermus aquaticus is a gram-negative, non-sporulating, rod-shaped bacteria with a length of 5 to 10 micrometers. Cells tend to form pale orange and yellow biofilms, which are composed of dense masses of rods and spheres. These rods have an odd ability to create spheres, currently believed to be through asymmetrical division. As far as diet, they are able to grow using multiple molecules. During nutritional studies, they were able to grow with glutamic acid as their only carbon source and NH4 and glutamate as their only nitrogen sources. They have transport systems that pair with ATP hydrolysis with the transport of amino acids, pointing to those being their primary energy source. During anaerobic conditions, pyruvate is formed through glycolysis and can be converted to acetate, lactate, and ethanol. While during aerobic conditions, a complete citric acid cycle is predicted, and they also perform arsenite oxidation, which points to the use of arsenic respiration. They grow optimally between a pH of 7.5 and 7.8, but there is no growth seen outside the range of 6 to 9.5. While at a temperature of 20 to 30 degrees Celsius, the bacteria had very little activity, and during the experiments, growth was so slow that one division took 6 hours to occur, while at their optimal temperature of 70 to 75, they grew so rapidly that a stationary phase was not seen, and after 24 hours, the culture reached its maximum density. But along with this rapid growth, there was rapid disintegration. Within 36 hours, they were back to their original cell count. Also, unlike other thermophiles and strains of Aquaticus, Y51 is able to survive in aerobic and anaerobic conditions. Thermus aquaticus was first discovered in the Yellowstone hot springs seen in the image on the left, and is what contributes to its distinct colors. Another recently discovered thermophile is TS, which was found 3.2 kilometers below ground in an African gold mine. They have also been able to fill other new man-made niches, such as compost manure and sewage sludge, which points to the versatility of the bacteria and other members of the Thermus genus. Overall, they function best at high temperatures and a neutral pH. And again, they are all able to live in aerobic and anaerobic conditions. The genomic plasticity is a major factor in the success of the thermophile genus and is mainly done through horizontal gene transfer. This was shown by similar composed genomic islands being found between the Thermus group. Within the genus, there is also a high rate of genomic rearrangements that led to the disintegration of functional operons. This could either be a problem as a result of environmental pressures or an adaptation process. By splitting the operons into smaller independent regulons, they may be better suited for their environment. Another part is their rapid reproduction. Once their conditions are optimal, they're able to rapidly divide and increase in population in just under a day. And this is likely a result of living in such extreme environments, although there is the trade-off, the rapid disintegration. The metabolic flexibility also plays a large role, being able to complete these citric acid cycle to produce energy along with other forms during anaerobic conditions, and being able to use multiple carbon and nitrogen sources. Before I start talking about phylogeny, I'd like to mention how the genome of Y51 was sequenced. Originally, a draft genome was made using both Illumina fragment libraries and Sanger chemistry. The 8 kilobase mate pair libraries and fragments were constructed using NX sequence, DNA sample prep, and log mate pair library kits. Final genomes were confirmed via PCR, and primer pairs were designed to validate and strengthen areas of low sequence coverage, similar to ours when we saw certain areas weren't strong or were messy within our sequences. Using a maximum likelihood method based on the 16S rDNA sequences, YT and Y51 were arranged as sister taxa, and TS was the closest related species. Comparing Y51 to SA01, a strain of TS, it was found that there were 90 conserved proteins between the two, 31 of them being related to ribosomal, translational proteins, and proteins involved in the electron transport chain. But there were 369 unique proteins to Y51. Then a separate study on the Thermus genus, there was an 80% homology to closely related lineages of TS, and 72% of the genes were acquired within the Thermus phylum. Bacteria play a crucial role in the ecosystems of hot springs, specifically the ones found in Yellowstone. Every year, geothermal fluids discharge 100,000 kilograms of arsenic into surface waters, increasing arsenic levels. The bacteria is able to convert arsenite to arsenate and does so at its highest rate during its growth phase. This is important due to arsenite being much more toxic than arsenate, and its ability to oxidize points to its ecological role in controlling arsenic bioavailability and toxicity in these environments. There's some really cool research based around Thermus aquaticus. There's a polymerase chain reaction amplification of alleles, or PASA, which can be used to detect alleles, mutations, and SNPs that cause genetic or infectious diseases by using modified DNA polymerase of Thermus aquaticus. There's also PNA, a DNA analog that is able to use TAC as a transcriptase when given a PNA template, and this has the potential to impact gene therapy, molecular diagnostics, and nanotechnology. And lastly, it can be used to track bacteria populations by cloning Thermus aquaticus 16S rDNA into bacteria and using high throughput sequencing to track the reads. Lastly, why do we care? Thermus aquaticus has played a large role in biological sciences due to the fact this is where we have discovered Taq polymerase, which has been used in many different sequencing techniques and has been used in medical testing for a long time, such as the PCR test for COVID. It also has uses for gene modulation and nanotechnology. If you have any questions, please feel free to contact me.